The second speaker is Dr. Sin, who is from the Department of Medicine at Stanford with a secondary appointment in the Department of Pediatrics. And Sin's interest and really passion has been to understand the mechanisms that regulate the development of the heart. And indeed, the ultimate goal of his research is to use stem cells from the heart to correct heart diseases, including heart congenital malformation. So, Sin, please come to the podium, and thank you for being here.
Great. I want to thank Dennis and the organizer for the invitation to speak, and also for Maria's for the kind introduction. What I'm going to do today is to tell you a little bit about the work that we have done looking at engineering an artificial heart. So first, what I want to do is to take you on a little bit of time travel back to 1550 BC, where it was discovered that the very first human prosthesis in an Egyptian mummy, and you can see this quite clever Egyptian found a way with the tools that was available at the time to replace the lost organ. In this case is the big toe. And now fast forward 3,500 years to 1990. In this cover from Science magazine, what you can see is that there are now quite a number of different prosthetic devices that are being used. So you can see artificial limb that Nadia has shown a little earlier, as well as artificial hip and also knee. And in the chest structures, what you can see, there are heart pumps and also the pacemaking devices. And the heart is an area that I work actively in. And what I wanted to do is to also tell you that there is a growing number of demand for organs, in particularly the heart, and that number is really just staggering because we have just an incredible limited number of organs that are available for transplant. And with that, I want to give a shout out to the Packer Hospital, who really has been the greatest transplant site on the West Coast, and also in the number of organs, it's the #1 in the country, and they really have just done fantastic work in this area. But obviously, we have lots of people with heart failures, and they really need to find a solution. And as you see, 115,000 people died of heart failure. So with this sort of staggering number and continue to grow, how can we actually find a way, to help treat this? So first, I just want to tell you a little bit about what, if we were trying to find a replacement heart, what we actually need. So the heart is the very first organ that is identifiable in development. And in fact, early on, Aristotle believed that the heart was even the seat for the vitality as well as intelligence and emotion as well as sensation. Now, obviously, in the thousands of years that's gone by, we have become much more in understanding exactly what the heart actually involves. And the heart is really remarkable because in the lifetime, it can beat 2.5 billion times, and it pumps 2,000 gallons of blood in every single day through 60,000 miles of blood vessels within the body. And this remarkable structure is made out of a variety of cells, including muscle cells, blood vessel cells, the electrical system, the conduction, and also valves that regulates the direction of the flow. So with something that's quite so complex, how can we actually try to make a replacement of this? Well, at least for now, what we have that is a sort of substandard replacement, although nevertheless, it's actually what is being used is a device called the left ventricular assist device. And this device is nothing more than just a mechanical pump. And so what you can see is that this device, which is implanted inside the chest of the individual, it draws blood from the major pumping chamber, which is failing, and it drives it through a propeller mechanism to push the blood all the way through the artery to then deliver it to the rest of the body. But as you can see, it's quite cumbersome. You need to wear battery packs because that's what's actually allowing this pump to function, and obviously you wouldn't want the battery to run out. And and there are wires that are coming from the chest out into the battery outside. So quite cumbersome. But, so why do we take on a cumbersome piece of a tool like this? Well, it's because people have shown that it actually increases survival. So one of the earliest study that was done called Rematch have shown that people who are implanted with these devices are able to increase their lifespan on average by 190 days. Which is really great if you're in end-stage heart failure, but obviously, as you can see, these curves eventually converge at about 2 to 3 years. We're a little better now than we used to be when this study was initially done, but these obviously are not permanent solutions because all patients, even with the device, eventually die. So why do they actually die? These devices are mechanical based, and they have extreme risk of having clot forming on top of them. So you can see foreign body inside the blood system, all of them will clot unless you provide blood thinner, and this is an example of what happened when one of these devices generates clot. You know, quite nasty, and on top of it, there are other issues as well. So in order to give the blood thinner, you're at risk of bleeding from the blood thinners that you provide. And also, they could still form clots and shower to the brain and cause a stroke. And in 30% or so of these devices, they have infection throughout the life of this device implant. So obviously, not the perfect solution, but this is the best that we have so far. So envisioning what might be a ways that we can generate bioartificial heart, some of the colleagues and collaborators in our field have been thinking about how can we generate a heart structure that would essentially mimic a normal functioning heart. So here's an example from one of our colleagues, Harold Ott and Doris Taylor, where they done the process called Decellularization, which is basically taking a heart tissue and treat it with detergent to wash away all of the living cells inside to generate essentially a hollow scaffold of a structure. And then what they envision then is to take muscle cells and blood vessel cells and re-implant into the scaffold structure. And here's an example on the bottom you see where they replace those cells. And then remarkably, they're able to show that in the cells which you replace with the muscle, they actually have begun some contraction activity, including the electrical activity as you see in the traces down below, like what you normally see in the human EKG. So, you know, quite interesting, but obviously these are done in animals, so a lot of, things will have to be developed in order to scale this up into human sizes. But one of the things I want to talk to you about, since we're in a stem cell session, is how can we use stem cell as the source to provide the muscle for this? So early on in my lab, we developed a mouse model where we can tag the heart muscles with a reporter that glows green when the heart muscle cell is formed, and we go through an in vitro differentiation process, so done entirely outside of the animal in the laboratory, and we generate muscle cells that can beat in the dish. So you can see these green muscle cells that we can find in the laboratory dish that really resembles quite well of what the normal functioning heart muscles. And now, as Nadia mentioned about the induced pluripotent stem cells, that we can derive the stem cell from any individual, from blood cells or from skin cells, and be able to then take that cells to differentiate into various heart-forming cells that we need, including blood vessel cells. So here's an example of the various different endothelial, which are the lining cells, the blood vessel, that can be made from these iPS cells. And obviously, since we're interested in the heart, it would be great if we can take these human-derived stem cells to make the contracted muscle cells. And so obviously, having the raw material for this, we began to start thinking about, well, how can we engineer these into a format that would actually be helpful in contracting? So with our collaborator from Harvard, what we initially generated was what we call diving board. And what you're looking at right now is that there is a backing of a scaffold material with the muscle cells on the underside of it. This entire setup is bathed inside a solution, and when we stimulate it with electrical activity, these muscle cells would contract, and it bends this diving board in solution and allow us to be able to measure with each contraction cycle how much force it's actually generating. And then what we could do is, with the force that we can measure with each contraction, figure out how many cells we're going to need for a certain amount of force that we need to generate. And our collaborator, Kit Parker at Harvard, also has done some really interesting work with these two-dimensional structure. And here you can see he puts the muscle on the underside, and on the left is a jellyfish, and in the middle is the engineered muscle. So lots of cool little things that you can do with these beating muscle cells. So these are done as two-dimensional structures, and obviously heart is a three-dimensional structure. What can we do to make these as three-dimensional organs? Well, we're certainly nowhere near being able to do that, but I did want to bring up something that is really interesting, and that is the use of 3D printing technology. I know now you probably have seen 3D printers are used in making all sorts of things, including food. So obviously, I think the future is to figure out ways that we can use 3D printing as a way to generate different tissues and organs. And here's some example of 3D printed structures that we have undertaken to using a scanning picture from a CAT scan. And we would like to advance forward to use 3D printer that could help us generate different structures, starting from the simple to eventually more complicated. And here's an example from Anthony Italia where they generated the scaffold for the ear and seed it with stem cell to regenerate something simple like the ear. And this is just one last thought for you, is whether it would be possible in the future that we might actually be able to do this to make the full-scale functioning heart. And we certainly hope so, and we're in the process of trying to think about ways to do this, and certainly would love to be able to do this in our lifetime. And thank you very much for your attention.